When functional medicine practitioners attempt to assess gut health, they almost universally rely on a single diagnostic tool, stool DNA sequencing. This method is designed to catalog the physical contents of the gastrointestinal tract. By reading the genetic material, these tests map the specific strains of bacteria residing in the colon. But relying strictly on genetic sequencing presents a critical biological blind spot. The mere presence of a bacterial strain does not guarantee that it is actually doing anything. In a complex ecosystem like the gut, millions of microbes can exist in a completely dormant state, while others are actively fermenting nutrients. DNA sequencing cannot differentiate between a microbe that is metabolically dead and one that is highly active. Traditional stool tests provide a highly detailed census of the inhabitants. They fail to measure the functional output of the microbiome, what those bacteria are actually producing on a daily basis. To measure functional output, we have to look past the colon and examine the bloodstream for circulating metabolites. The gut and the systemic immune system are not isolated silos. They are engaged in constant bidirectional communication, using these microscopic metabolic byproducts as their chemical language. We can now intercept this communication using a dried blood spot method. By applying high-precision liquid chromatography, specifically HPLC UV FLD technology, we isolate and quantify these exact signaling molecules. Because circulating metabolites respond to recent food intake, capturing an accurate baseline requires biological stillness. The test is performed after a strict 10-hour, water-only fast to eliminate acute post-meal interference. By analyzing specific interacting metabolites in a single drop of fasted blood, we extract a precise mathematical read on how the microbiome is functioning. This diagram maps the primary precursor material for gut immune signaling, an essential dietary amino acid called tryptophan, or TRP. Once absorbed from your diet, tryptophan acts as a highly contested metabolic crossroad. Based on the internal environment of your body, TRP is forced down one of two distinct pathways. In the first route, beneficial gut bacteria ferment and convert tryptophan into a protective metabolite called indole 3-propionic acid, or IPA. High circulating levels of IPA yield specific physiological benefits. It actively reinforces the integrity of the gut barrier and provides systemic protection against oxidative stress. The alternative route is driven by the host. Systemic immune cells hijack the tryptophan supply, converting it into kynurenine, or KYN, through the kynurenine pathway. This diversion is often triggered by increased immune stress. When the body's immune load rises, tryptophan metabolism shifts into the kynurenine pathway, reducing the substrate available for beneficial microbial output. The fate of your dietary tryptophan reflects a metabolic fork. Whether your system is currently prioritized for gut protective homeostasis or systemic immune stress. This dashboard displays how we translate the tryptophan crossroad into three measurable blood ratios. The first metric is the IPA to TRP ratio. This isolates your gut's mechanical output. It measures exact microbiome efficiency, calculating what specific proportion of dietary tryptophan your gut bacteria successfully converted into protective IPA. The second metric is the KYN to TRP ratio. This acts as an established marker of immune activation, quantifying the systemic stress load diverting resources away from the gut. The final metric is the IPA to KYN ratio. This scores the relative balance between your protective microbial pathways and your stress-driven immune pathways. These ratios remove reliance on subjective symptoms. They provide an objective mathematical index of the competing pathways between your gut bacteria and your immune response. To drive the tryptophan pathway back toward beneficial IPA production, you have to feed the specific bacteria responsible for the conversion. The required fuel source for these highly specialized microbes is complex prebiotic dietary fiber. Yet standard dietary interventions often fail to correct dysbiosis. The flaw lies in the fermentation speed of conventional single-fiber supplements. If you consume only one type of fiber, like standard psyllium or basic inulin, it will ferment entirely too rapidly. The fuel is completely consumed in the proximal or upper colon. As a result, the bacteria residing further down in the distal colon are left starving. Without substrate to ferment, they cannot synthesize indole-3 propionic acid. Conventional single-fiber products fail to resolve systemic dysbiosis because they lack the kinetic diversity required to physically reach and nourish the entire organ. The solution is to engineer kinetic diversity using a targeted multi-fiber approach. This chart shows the composition required to achieve full bowel fermentation, a matrix of 8 distinct naturally derived dietary fibers. This specific formulation spans a variety of complex structures, pairing resistant starches from corn and green banana with oat beta-glucans and chicory root inulin. This timeline illustrates the mechanical logic of the matrix. Because each of the 8 fibers features a different molecular density, they yield a staggered, time-released fermentation. As the bolus moves through the digestive tract, different fibers begin their breakdown cycle. This ensures a steady fuel supply reaches the beneficial IPA-producing strains located across the entire large bowel. Structural diversity in the prebiotic input provides the physiological framework for consistent metabolic output across the length of the large bowel. Forcing fermentation across the entire bowel extends well beyond correcting the tryptophan pathway. Deep colon fermentation acts as a mechanical trigger for the direct stimulation of GLP-1, a critical endogenous gut hormone. Instead of relying on synthetic peptides, this bacterial action triggers your body's GLP-1 release, which regulates appetite and normalizes systemic metabolic signaling. Furthermore, the specific components of the matrix serve secondary metabolic functions. The inclusion of resistant starches works to physically blunt the sharp rise of blood glucose following a meal. Simultaneously, the specific beta-glucans sourced from oats bind to bile acids, performing a scientifically recognized role in maintaining normal blood cholesterol levels. A diverse fiber matrix targets both the restoration of gut bacterial pathways and the activation of the body's broader metabolic and weight management networks. Physiological optimization requires a shift from generalized observational data to functional blood metrics. The process begins with a quantitative baseline, measuring your circulating tryptophan metabolites in the blood. You then deploy the 8-fiber prebiotic matrix to systematically alter your fermentation kinetics, providing nourishment to the entire microbiome. To measure the impact of this targeted nutritional protocol, the cycle must be maintained for 120 days. After 120 days, a second blood test is required. This objectively verifies the biological shift, providing mathematical evidence of how your system has moved from KYN dominance back toward a protective IPA dominant state. This 120-day protocol transitions the approach to gut health from generalized supplementation to a cycle of objective measurement and targeted functional nutrition.